So I had 105 patients in the study that was enrolled, and 6 of them had beta thal and sickle cell. So we kind of didn't include them in our study because we wanted to only focus on people living with sickle cell disease. So this would be this would add kind of like a bias in the research, right? You want to focus on one population, otherwise your data will, not look as good. So yeah, 99 patients, and basically we looked at magnetic resonance angiography. Has anyone had magnetic resonance and angio here before to look at kind of their blood vessels in sickle cell disease, beta thal? No one has had any experience. Yeah. So what they do is basically they kind of put things like a little machine in your temple. It's kind of like an ultrasound, and they look at every single blood vessel, major, minor, that goes to your brain. Okay. And if they see any blockages, it's considered an abnormal MRA, right? So we grouped people who have abnormal MRA and stroke together because an abnormal magnetic resonance angiography test can lead to strokes, right? So it's a defect. So we grouped those together, and patients with no abnormal MRA and no stroke, we group them together, okay? And so let's go on to the results. Okay, so this is the first graph is an association between hemoglobin F. So on this Y value here, right, you have hemoglobin F levels, and then in your X value here, you have either the absence of the XMN polymorphism, right? We want the presence. So the presence of that T increases your hemoglobin F values. That's what we're trying to see, right? And basically, how you see in science that your results are significant is they have to have a 95% confidence interval, right? Because it has to be representative to all populations. So we found this to be very, very significant, that people just with this one SNP, right? And this is only people who are heterozygous for that SNP. Remember, we talked about there's 2 genes. So this is only one of them changing from a C to a T. They had a 3.4% increase in fetal hemoglobin F, right? Even 3% increase in fetal hemoglobin F is significant, right? So the average basically of people with this XMN polymorphism was around, what did we say it was? It's around like 12%. That's the average, about like 12, 13%. And then if you had an absence of this polymorphism, your hemoglobin F levels were about at 7%. Sorry, that's actually like 10. So there was a 3.4% difference. So I think these are the correct values. So 10.85%, right, if you have the SNP, and 7.5% was your baseline fetal hemoglobin value. So 3% increase on average with just one SNP. And there's so many different SNPs that you can have. There's SNPs, there's about 20 to 30, I think 20 SNPs, these polymorphisms that are associated with fetal hemoglobin F levels, right? But do people have them? That's the question. If people have them, it's protective, right? If you don't have them, people usually have more severe disease with sickle cell. So, so as I said, right, stroke manifestation is not known why it occurs in people living with sickle cell disease. So I wanted to see if I can find an answer with one of the SNP that I was working on. So I kind of wanted to see if this SNP, this is, okay, I think this is the same graph actually. So this is wrong. Okay, sorry about that. But so what I did was I had a graph that was abnormal and normal MRA. So people who have stroke, and I tried to see if it was correlated with the hemoglobin F levels, and it wasn't correlated. So that, which means that people with higher hemoglobin F levels still had stroke, right? So there was still kind of that disparity. Why do people with a higher fetal hemoglobin, right, if they have less occlusive episodes, why do they have stroke, right? And that's kind of the bread and butter, like why does this happen? And it's still a kind of like a mystery, and there's more research going on in that field. Okay, so the conclusions of the study, right? So when people are born with sickle cell, right, there's so many of these SNPs that we're talking about that could either help them, right, clinically, or it won't help them at all, right? And they'll have more severe disease. So can we, when a person is born, can we do genetic testing and find all these genetic variants that increase hemoglobin F, right? And can we see if these infants, these babies, are going to have severe disease or less severe disease, right? This is really important because we have to see, as soon as these babies are born, we have to see if they're high risk people, right? If they're high risk, do they need chronic transfusions? Do they need hydroxyurea, right? Hydroxyurea is a medication that increases fetal hemoglobin values. So this is the main medication right now for sickle cell disease, is to increase fetal hemoglobin values, since the normal hemoglobin is defective, right? So that's kind of what it is. And I want to show you guys kind of this right here, okay? So for thalassemia in 2015, there was a research done in Canada where they identified all these SNPs, right? These genetic variants that increase hemoglobin F values. And you could see, basically you could test for you could get genetic testing, right? They'll see if you have these SNPs. So this is a SNP that increases hemoglobin F. This is a SNP that increases hemoglobin F. This and this, these are all genes. Okay, let me rephrase that. These are genes that if they have a T for this one, right, you'll have higher fetal hemoglobin F. So you do genetic testing, you get your results, okay, for these genes. Okay, you say, okay, I have a T, okay, and I have GG, I have AG, and I have CC for this, okay? And then I have, let's say, mild to severe beta phenotype, okay? So this is, let's say severe. So this is severe. Severe is basically beta thal major, okay? And then if you have a gene in an alpha gene, you could put that. So this is for each patient, right? Depending on, and let's say I'm a male. You can calculate, no deletion in the alpha gene, okay? You can calculate the disease severity, and it will show you a graph like this, okay? That what kind of severe that you're going to have. So with taking everything into effect, it says that you're going to have low severity clinical outcomes, okay? So there is no such thing done for sickle cell disease. There's something done for thalassemia, right? So this is important when babies are born, right? We want to see if the babies are going to have less or very severe sickle cell disease outcomes, okay? So this is for thalassemia. They made one for thalassemia, but they haven't made one for sickle cell anemia, which kind of sucks. So this is the thing. I want this to kind of happen. This is, I think, very important for people living with sickle cell disease as soon as they're born, kind of to help their clinical symptoms out. Okay, so I just wanted to share that with you guys. This was supposed to be the goal of my research, because I had also these SNPs that were done by the doctor Carolyn Hoppy. So she actually for the same patients, she got all of these SNPs and she made a whole data chart, but she left to Global Blood Therapeutics, so we weren't able to complete the study. And I really wish that we could have completed the study. And I tried so hard to find another researcher that could help me with the statistical analysis, because that's very, very hard. And it gets into a lot of computer science that I have no idea. So that's what I really wish that I could have done with this study. So hopefully I'm still trying if someone, can help me kind of figure out this data and how I could present it to the sickle cell world.